Hey biology, so tonight we're going to do a couple different things. We're going to talk about chapter 7.1, focused mainly on the difference between prokaryotic and eukaryotic cells, and then the cell theory. You will be responsible for knowing the light microscope and the different parts of the light microscope, so be sure to study that. We are going over that for 3 days, or using the scopes for 3 days, so they are important. So we're going to go over basically the cell theory, and then we're going to talk about the cellular organelles, and we're going to go ahead and fill in the sheet here. So this is your coloring sheet. We'll talk about all the functions, and then you can make sure that as we're talking about those functions, that you, if you don't have them written down, or you don't have them within your cellular organelle chart, that you get them within your chart or into your chart, so that we stay nice and organized, and that chart will be helpful for you when you're studying. Alright, so the first thing we're going to go ahead and do, we're going to look over just a few slides here. So obviously this chapter is over cell structure and cell function. So this is our cell, particularly an animal cell. All cells are going to be made of molecules. So we know that molecules, or hopefully you know from chapter 2, that molecules are made from atoms, and that's why we study atoms. So they are the smallest thing that biologists study is the atom. So again, like we said, atoms make up molecules. Molecules are what make up our organelles. So when you hear that term organelle, what you want to think is about organ, and so organ is in the term. So organelle is basically similar to what organs do in our body. Organs have different functions and perform those different functions, but as a whole, they perform functions that make our bodies work properly. And that's going to be the same thing for the organelles within a cell. The organelles will perform different functions, but all together or collectively work together to make the cell function in the way that it needs to function. So when we start talking about the cell, we need to mention the cell theory. So the cell theory basically has 3 components to it. And the first one states that all living things are made of cells. And if you don't have this somewhere in your notes for your 7.1, make sure it's in there and highlight it. Cells are also the basic unit of structure and function in an organism. So when we say that it's the basic unit, we just mean it's the basic unit of life. The last part of the cell theory is that all cells are produced from existing cells. So a cell just can't appear out of nowhere. It's going to be produced from another cell. So that's via asexual reproduction, where a cell will just divide itself, or it's via sexual reproduction, where 2 cells come together to make one. So we have different types of cells. We have plant cells, animal cells, and bacteria cells. And these cells can be grouped into 2 different categories, and you need to know these 2 different categories. So the 2 different categories are, so the first set are cells that don't have a nucleus. So the nucleus is like the brain of the cell. So it's basically the organelle that contains all of our genetic information, so all of the important instructions to life. So in these types of cells, we have no nucleus. We still have the DNA, but we don't have the membrane that surrounds that DNA. Okay, so we don't have membrane-bound organelles. And these types of cells are called prokaryotes. And whenever you hear that pro, that means like before. So this is before things got complex. And when we think about those cells, or an example of that cell is a bacterial cell. So bacterial cells, even though they're not very complex, they've been around for a long time. Okay, so bacteria cells have no membrane-bound organelles. Their DNA is kind of just jumbled up in the center there. Alright, and then the other cells are more complex cells, and we group plant and animal cells in here. These are going to be the cells with nucleus and organelles surrounded by membranes, and we call those eukaryote or eukaryotes. And the way for you to remember maybe this one is, it sounds like eukaryotes, it sounds like you, so it's your cells. Your cells are like this, so your cell plus a plant cell are going to be like this. Okay, so the next thing that we need to do is we need to go ahead and get this sheet out in front of us. So get it out in front of you. It should already be glued into our notebooks. We're going to go ahead and color it, and I'm going to talk about the different functions. So as I talk through the functions, make sure that you have the proper things or the correct things filled out within your organelle chart. Okay, you guys, so now that we have this in front of us, we can see that we have the three different types of cells that you are responsible for knowing. We have our category, the prokaryotes. So let's go ahead and write our example here. So I told you an example of the prokaryotes earlier in the video, so tell me what that example is. So what is this an example picture of? When we say eukaryotes, we know that we are talking about the animal and we're talking about the plant cell. So remember, those are your cells, okay? So why don't we go ahead and write what makes a eukaryotic cell a eukaryotic cell, okay? So I went over that earlier, so go ahead and make sure you write that in right there, take a few notes. Alright, so the first organelle that we are going to talk about is going to be found in all three of our cells. And that is going to be, and that's this category here, that's going to be the cell membrane. And I would like you to go ahead and color the cell membrane in pink. Okay, so the cell membrane is here. Now when we're looking at our cell diagram, the cell membrane is going to be this part right here. So we're imagining that this is a structure that's been cut apart. So all of this is a cell membrane. This is also part of the cell membrane. So this right here. Okay, so we're going to want to get all of that in there is going to be pink. Okay. Now the way that you could tell that this is an animal cell and this is a plant cell is basically its overall shape. Obviously there's going to be some organelles in an animal cell or some organelles in a plant cell that are specific to that cell. However, the shape can help you tell as well. So this one is more circular in shape, whereas plant cells are going to be either more, they're going to be more rectangular or square in shape. So inside of a plant, the cell membrane is going to be found on the inside of the cell wall. So this little lining here is considered our cell membrane. Okay, so you're going to go all along and color the inside membrane there. And that's the cell membrane. Okay, the cell membrane in the prokaryotic cell or the bacteria cell is just going to be all the way around. And so you can kind of color that in here. Okay. Color that all the way around. Okay, make sure you color the whole thing. I'm just doing this as an example so that we don't take up too much time. Also, if you want to just pause it as we talk over things or you want to get things colored in as we go, that would be probably a great idea and easy for you to do. Alright, so let's talk about the cell membrane. Remember, as we're talking about each cellular organelle, you want to make sure that in your cell organelle chart you have the correct information. So the cell membrane we also call the plasma membrane. They're made out of predominantly two things. They're made out of one, phospholipids. So basically that's a lipid. So we have all of these little circular plus these two little tails there. So we have the glycerol and we have the fatty acids here. And we have proteins. So proteins are obviously these purple things here that are labeled proteins. Okay, so lipid tails are hydrophobic. If you remember hydrophobic, we hate water. So hydro means water, phobic means phobia, it's water hating. So there's our hydrophobic glycerol end. Okay, or hydrophilic, sorry. There's our hydrophilic glycerol end, and that's going to be the end that will be able to touch water. So that's going to form like the outside layers of our cell membrane. And then our fatty acids are going to be our hydrophobic ends. They don't want to touch water, so that's going to be found on the inside of the cell membrane. So water and oil don't mix. So right here we have some phospholipids. Basically, our oils are phospholipids, and what happens if we throw a bunch of phospholipids into water? Well, they will either form this little circular like molecule here, or they'll eventually just separate so that the hydrophilic ends are touching the water, hydrophobic ends are not touching the water. So what we have is we'll form in our cells a phospholipid bilayer, where we have this part here, this inside part is touching the water in the center, and then this outside part is touching the water as well. But then we have this area here, okay, this inside area where we have our hydrophobic tails that are not touching that area at all. So our cell membrane, it basically functions to help things move in and out. So the major function of the cell membrane is to allow things in and out, or the proteins. So proteins, if there are substances that are too big, so for instance this sodium substance too big, we're going to use a protein to let it in. So again, what does it do? It's going to act as a boundary. It's going to control what enters and leaves a cell. So an example, if we were to do like a cell analogy project, it's the gatekeeper. It's going to allow things in and out. It's going to keep foreign substances out, let food and other things in, get rid of things that the cell doesn't need. All right, let's move on to our next organelle. So let's find it. Okay, so our next organelle is going to be the DNA. DNA is going to be found in all three of these things. So DNA is right here. And DNA is going to be found inside of the nucleus. And let's see if we can, there's me. Okay, so DNA is found all inside here. All this squiggly junk here. It's going to be found inside of the nucleus in the plant. Right here. And it's going to be found not as chromosomes inside of the nucleus here, because there's no nucleus in the prokaryotic cell. Remember, there's no cellular bound organelles in that. So DNA is going to be right here in the bacteria cell, not surrounded by a membrane. Okay, so we know that the nucleus is, or the DNA is found in the nucleus, and I hope you know by now, after talking about it, that DNA is basically our nucleic acid, so deoxyribonucleic acid, that basically codes for all of your genetic traits. And so in a plant and animal cell, it is kept nice and safe in the nucleus. So let's go ahead and color the nucleus. Now you're going to notice that there's no nucleus in a prokaryotic cell. And the reason why there's no nucleus is because there's no membrane-bound organelles. Remember that this pro means that it's before, so it's before the nucleus. So prokaryotes, most simple. So let's go ahead and color our nucleus this like purple color, and we're going to color basically all parts of the nucleus that purple color. So we have the nuclear envelope, so that's basically just the outside of the nucleus. We have the nuclear pores that allows things in and out, and we have the NUCLEOLUS. Okay, so all right there. So in the animal cell, it's going to be our largest organelle here, and it's going to be kind of in the center. So this right here is the nucleus. Now the little holes right there, those are the nuclear pores. We can kind of darken those if you want. So those are a little bit darker. And then we have the nuclear envelope. That basically is what surrounds the membrane that surrounds the nucleus and so forth. So that's in the animal cell. In the plant cell, it's not so much in the center because we have that large central vacuole, but it's going to be right over here and look very similar to the animal cell nucleus. Alright, let's see, we'll color this, circle around these, and there we go. So that is our nucleus. So the NUCLEOLUS is what's in the center here. So we find that in the center. Why don't we darken that one up too? Alright, so let's go ahead and talk about the function of the nucleus. Hopefully you know that basically the nucleus is the brain of the cell. In an animal cell, it's the largest organelle. It's going to be found in the center. Okay, so the nucleus is surrounded by a nuclear envelope. So similar to our cellular membrane, we're going to have a nuclear membrane where we have basically a phospholipid bilayer, so hydrophobic, hydrophilic layers. And it's a double membrane. So we call it the nuclear membrane. We have nuclear pores, and that's basically, like I said before, it allows molecules to move in and out of the nucleus. So what does it do? It contains our genetic material. Now, our genetic material is very important. It codes for all of the enzymes in your body, which enzymes are made of proteins. So it codes for all of the different proteins that you have in your body. It keeps it safe. If you damage the original set of DNA, then you pretty much damage that cell, and it can't be repaired. So it's going to keep basically our DNA safe. So it's the control center of the cell. Like I said before, it's the brain of the cell. So it has all the genetic code, and the genetic code tells the cell's parts what to do. So it's like the dictator of the cell. The nucleolus, so that dark part in the center that we colored, it's going to basically make RNA for ribosomes. And we'll get more into ribosomes when we start talking about protein synthesis. But RNA, if you remember, is a nucleic acid it's single-stranded, and it makes ribosomes, and RNA also helps in the production of protein. In our diagram here, we are going to go ahead and focus mainly on the animal cell, and we're going to color these organelles orange. So these are the centrioles. So the centrioles we will only find in animal cells, and centrioles are going to basically be these guys right here. So those are our centrioles. You will have to be able to look at a picture. You've already drawn it. We're coloring it in now, but you will have to recognize what the organelle looks like and what each one is. So you have to know picture, function, and obviously name of the organelle. So we have one set here, and that's it. So what do our centrioles do? Centrioles are going to be important for, let me pull this up, okay, for cell division. So when we're pulling apart chromosomes during cell division, so you might have talked about mitosis last year, but in mitosis, when we go ahead and make new cells, we have to copy our DNA and then pull apart the chromosomes so that each new cell has identical sets of chromosomes. Well, the centrioles help in that. And so here's just an example. Our chromosomes will eventually line up in the middle of our cell. The centrioles send out these fibers or meiotic or microtubules that then pull apart the chromosomes. Alright, so our next organelle is the mitochondria, or mitochondrion, and then plural, mitochondria. So we're going to find that in our handy-dandy diagram. Mitochondria is going to be found in plant and animal. Well, when you think about it, why is it found in both plant and animal? If plants have the ability to make their own food, why do they need mitochondria? Well, the reason why plants need mitochondria is because they make their own food. So they use chloroplasts to make sugars, but then they have to go ahead and take that food and they have to break it down into energy that's usable for them. So we're going to find mitochondria in each one of our cells. In the animal cell, we're going to color the organelles that look like this green. And I'm kind of regretting that I colored that green, but we'll color the chloroplast a darker color green. And so they're going to be like these bean-shaped little organelles around the cell that basically function in the production of energy. So let's go ahead and take a look at the mitochondria. Okay, so they look like little sausages, okay, or I like to say little jelly beans. They're surrounded again by a double membrane. It has its own DNA. It's actually a very complex organelle. The folded inner membrane increases surface area. Very important. Make sure you know that. It will come up later in this semester, in this semester. So it basically has a double membrane inside. The more folds it has, the more chemical reactions it can do, and the more energy it can make. Okay, mitochondria come from the cytoplasm in an egg cell. You inherit your mitochondria from your mother, which is pretty interesting. What does it do? So it's the power plant of the cell. It burns glucose to release energy, and it stores energy as ATP. So if you remember from last chapter, ATP is a nucleic acid or a nucleotide that basically gives us little small bursts of energy. It's basically our energy currency. It's what we can kind of burn really quickly. Alright, so our next organelle is going to be our cytoskeleton. The cytoskeleton is found in both plant and animal, but we see now it's found in our prokaryotic cell. It's going to be found in all of our cells, so let's go ahead and color cytoskeleton. We'll color that blue here. Cytoskeleton is basically going to be like the skeleton for the cell. It's going to allow the cell to keep its shape. So we have these little tubules here these little squiggly lines here, and anywhere you see those things, that is going to be your cytoskeleton. And we're going to see the same thing in our animal cells. So again, it helps it keep its shape. Alright, so in our slides, we would have right here, just similar to what I said, help cells maintain shape, help cells move organelles around. So if we need to move vesicles when making proteins from one place to another, we'll use that. So it's made out of these proteins or microfilaments and microtubules. Don't worry so much about those names, not as important. So our next organelle that we are going to talk about is also found in all three of these things. We are going to go ahead and label this red. We're going to talk about the ribosomes. And you know what? Microtubules and microfilaments, we're going to go ahead and label those blue as well. And you know what? We're just going to keep them blue because that's something that you'll learn in Bio 2, the difference between the two, or you'll be asked to distinguish between the two. So ribosomes, we're coloring red. Ribosomes are going to be all those little dots that you see either on the endoplasmic or you see them freely floating in the cell. So all the little black dots, you would just go ahead and so they're pretty much black dots. You would just go ahead and mark red. So do as many as you can. If you miss a few, I'm not going to yell at you because there are tons of ribosomes. And you're going to find them here. So here is where they are in the plant cell. Boom. Just grab a few of them. We also, like I said, you're going to find them in the ER. So here's some. Okay, and then we're also going to find ribosomes in the prokaryotic cell, and those are those free-floating ones here. Okay, ribosomes are actually super important. As we already talked about, ribosomes are made out of RNA, so the ribonucleic acid, so a nucleic acid, and they're going to be made in the nucleolus of the cell. So if you remember, this is the nucleolus here, and then this is the nucleolus. Alright, so ribosomes, when we talk about them, are made of proteins and RNA. It's basically, and for the function, it's the protein factory for the cell. So basically the ribosomes are what join amino acids. So if you remember, amino acids are the monomers or the building blocks that make up proteins. So the ribosomes actually join those proteins together, or the amino acids together. So they can be attached, like I said, either to the rough ER or free in the cytoplasm. Organelle is going to be the ER, and the ER is going to be found in, that's interesting because it says that it's just, so plant and animal. Yeah, I don't know what I'm thinking. It's going to be found in the plant and animal, but not in the prokaryotic cell. We'll color that pink. So we've used a pink before. If you want to shade in it darker, if you don't have more than one pink, that's fine. But we have a rough ER and we have a smooth ER, and they're different, and the reason why they're different is because they have different functions. So remember, structure follows function. So this right outside of the nucleus is our ER. So we'll just go ahead and color it in. It's basically these flattened, like little tunnels out here. So that's all the ER. So we have the rough ER, that's the one with the ribosomes all around it, and we have the smooth, find the ER here. Okay, we're going to find one here. And then, I don't know if I really see a smooth ER. Here's a smooth ER right here. Alright. This picture is not typically the nicest picture because we're going to, for the most part, find the endoplasmic reticulum next to the nucleus. And there's a reason why it's next to the nucleus, and you'll find out as you take notes, and we'll talk about that in a moment here. Okay, so the ER, let's talk about that. So we've already talked about ribosomes. Ooh, went a little fast there. So the ER stands for endoplasmic reticulum, basically a network of hollow tubes. Like I said, there's two kinds. We have the rough or we have the smooth. So the rough one, there are ribosomes on it. So when you think of rough, and you should think of ribosomes, rough ribosomes, okay? The ribosome's function is to make proteins. So basically the ER is made out of membranes, and the membranes are used to export proteins out of the cell. So it makes membrane proteins and proteins for export out of the cell. So basically, after proteins are made on the ribosome, the endoplasmic reticulum does some shipping to then the Golgi apparatus. So the rough ER has ribosomes attached. Proteins are made on ribosomes, inserted into the ER to be modified and transported. So for your organelle chart, you should have modify and transport proteins for rough ER. The smooth ER is going to be a little bit different. So the smooth ER won't have any ribosomes attached, and it has enzymes that perform special tasks. So the big thing that the smooth ER does, it makes membrane lipids. When I think of lipids, you should obviously think of fats because we've talked about it. But what you can also think of, lipids are smooth. So the smooth ER functions in making lipids. So, or like, I don't know, when you think of fat, you think of like smooth objects or like, you know, fat animals. They're usually quite like just, they're flubbery and they're soft and squishy. So basically a smooth ER might be smooth looking and squishy looking. And so it's going to make the lipids or the fats. So some more things that it does, it regulates calcium in muscle cells and destroys toxic substances in liver. Golgi apparatus is not going to be found inside our prokaryotic cell because it has no this has no membrane-bound organelles, but it's going to be found in both plant and animal here. So let's go ahead and color it a dark green. So I'm just going to press harder on this color here. If you don't have a dark green, there we go. There's Golgi apparatus. Made it a little bit darker. You can kind of shade in lighter or darker. You will have to know them. So if you have multiple colored greens, go ahead and use them. Just know that Golgi apparatus, and we can even point to it if that helps. So when I point to that being the green part right there. So this right here is Golgi. This is the organelle that functions like UPS. Okay, so it is going to basically deliver, like in the plant cell, might be the only one that they identify. Yep. All right, so let's quickly talk about that. So we have our Golgi apparatus. We also call it Golgi body, so I'll refer to it either or. You should just know when you hear that Golgi word that it can be both of them. It's all the same thing. So basically the pancake-like stacks. This is important because these modify, sort, package molecules for ER for storage or transport out of the cell. So like I said, this is going to pick up proteins. It's going to pick up lipids that are sorted and fixed inside of the endoplasmic reticulum and take them to wherever they need to be in the cell. Alright guys, so we're almost done here. Okay, so let's go ahead and move on. So now let's talk about the lysosome, and I'm going to color that with a highlighter yellow. The lysosome is basically the garbage factory of our cells. Okay, it's going to contain enzymes that allow the cell to basically digest, or let's say the cell becomes unable to function anymore. And so basically the lysosome will explode and the cell will perform something called apoptosis, which is basically like cell suicide. It destroys itself for the well-being of the entire organism. So lysosomes, when it comes to this, lysosomes are probably one of the hardest ones to actually like pick out and differentiate between lysosome and vacuole. So let's go ahead and just like find, let's just consider these right here in the animal cell lysosomes. Okay, so those are, again, the garbage bins of the cell. And then in the plant cell, any circular object here. So obviously, I'm not going to be able to tell you or point out a circle in here, a sphere inside of each cell and be like, what is that, the lysosome or the vacuole? That would be ridiculous. However, in a plant, you should know the difference like between a lysosome and a vacuole, because a vacuole in a plant, and I'm going to, I don't have any blue, let's see. Here's blue. So I'm going to color in the vacuole blue? Because the vacuole is going to store water and minerals. And so in a plant, the vacuole has a really important function. The vacuole is going to be central. So this whole thing right here is vacuole. Okay, and it's super important because the vacuole is going to go ahead and apply pressure to the plant. Because you know, our plants have to be rigid and they have to maintain their structure because they don't have the ability to move. And so having a large central vacuole allows there to be pressure on the outside walls, and it allows it to help keep its shape. So the cytoskeleton helps it keep its shape, but also the central vacuole, which again holds water and minerals, okay, allows it to keep its shape as well. Now in an animal cell, vacuoles, hard to distinguish difference between them and lysosomes. So we're just going to color some of the little spherical guys in here because there's just a bunch of them. We're going to color them blue. So in an animal cell, vacuoles, they have the same function. So they store water and minerals, but they're smaller and not central like the plant cell. Alright, so it looks like we only have a couple more things left to do, which, woo, that's awesome. So our next one, let's go ahead and we'll grab the orange again. So inside of our bacteria cell, our prokaryotic cell, we have a cell wall. And what makes it different from a plant cell is the fact that it is made out of peptidoglycan. Now, you don't have to know this. Don't worry about this. You won't have to know it until second semester. So let's go ahead, and this though is going to be on the outside. So a cell wall is always going to be on the outside of the cell membrane. So we're just going to shade around here. So prokaryote organisms have cell walls. And again, cell walls help cells maintain their shape. So plant cells, okay, they are going to need this like really stable structure that helps it keeps its shape. So in a plant, the cell wall is going to be made out of the cellulose, or out of cellulose, which, as you know, or hopefully you remember from last chapter, cellulose is a carbohydrate that plants use to store monosaccharides as. And we find it in basically our cell walls. So the outside here and the outside here of the plant, so all the way around, is going to be the cell wall. So basically, it helps support the plant cell. Only found in plant cells, not in animal cells, because animal cells have to be able to move. They have to have a fluid structure, and the cell membrane allows for that, but a cell wall does not allow for a fluid structure. Alright, so we are almost done. The next one, let's go ahead and take our light green and color our chloroplasts. Chloroplasts are only found in the plant cell. Chloroplasts are very important, okay? They allow the plant to capture the sun's energy and go through photosynthesis in order to make sugar. So here are chloroplasts. And here's another one. There. The way you can tell the chloroplast from a mitochondria is basically the chloroplasts have these little sphere-like discs inside of them. They're called thylakoids. You don't have to know that for now, but they have these little spheres, and those spheres basically help in part of the reactions or the different reactions of photosynthesis. So we have another one here. So they're both, it's bean-shaped, similar to the mitochondria, but chloroplast has little like sprees in them, whereas